Scientists took a long time to decide what is the genetic material in the living cells. We know that DNA acts as genetic material in all living cells except except viruses. DNA acts as genetic material. So what is genetic material? Genetic material is basically the most important component of a cell. It controls all the activities of a cell as well as reproduction of the cell, that is replication, DNA replication as well as replication of the cells. We know in DNA in case of eukaryotes it's present in the chromosomes, and in case of prokaryotes the genetic material is exposed in the cytoplasm. Now what we know that DNA is made up of genes which controls transcription, that is synthesis of mRNA, as well as translation, that is protein synthesis, which controls all the activities of a cell in turn. Now what are the experiments done? Name of the experiments. Name of the experiments. Basically three most important experiments. First one done by Griffith, that is transforming principle. This is done by Frederick Griffith. Second one, testing the biochemical properties of the macromolecules present in cells. And third one, that's the deciding one, Hershey-Chase experiment. Hershey-Chase experiment, which finally told us that DNA is the genetic material. Now coming to the first one, that is transforming principle done by Griffith. Transforming principle. Griffith done a series of experiments with Streptococcus pneumoniae, that is a bacterium causing pneumonia. I repeat, Griffith done a series of experiments using Streptococcus pneumoniae, pneumonia, which is a bacterium and it causes pneumonia. Now what he did, there are mainly two strains of Streptococcus pneumoniae found, that is smooth S strain, S in bracket smooth strain. They form smooth and shiny colonies when grown in a culture due to presence of mucus outer layering, mucus that is made up of polysaccharide. Mucus coat made up of polysaccharide. And they cause pneumonia. Another strain, that is R strain or rough strain, they do not have R in bracket rough strain. They don't have mucus coat. That's why they are called rough strain, and they do not cause pneumonia. So what he did, first he infected mice with rough strain and with smooth strain. So what happened? In case of smooth strain, the mice died. I repeat, let me write. So S strain plus mice died. Just a moment, I'll write. Mice died. In case of rough strain, the mice lived. R strain plus mice live. Living mice lived. Because we know that smooth strain can cause pneumonia. Now Griffith killed S strain. He'd killed S strain, so it won't cause. So it was unable to cause death in mice. Now what he did, next step, then what he did, he applied heat killed S strain plus R strain plus mice. Now this S strain is heat killed, this R strain is live. As a result, again the mice died. So Griffith decided there is one property. There is the S strain somehow transformed its properties to the R strain. S strain somehow transformed its properties to R strain. Now the next experiment, and we all know that transformation means transferring genetic material from one cell to another without forming any tube-like structure, means direct contact in case of bacteria, basically in same species. Now testing biochemical properties of macromolecules present in the cells. Avery, McCarthy and MacLeod, as you can see, Oswald Avery, Colin MacLeod and Macklin McCarthy done this experiment. What they did, they isolated purified form of macromolecules present in the cells of both R strain and S strain. They isolated proteins, DNA and RNA. Actually they done experiment related to the transforming principle experiment done by Griffith. Then they found that in absence of purified RNA or purified protein, the transformation property is still, the transforming principle is still applicable. They also, how they did it, they introduced RNAs, that is RNA degrading enzymes, and proteases, protein degrading enzymes in the cells of S strain as well as R strain. This in this experiment, this one, still they found that the transformation occurs and the mice died. Now coming to the most important experiment done by Hershey and Chase. This experiment finally decided that DNA is the genetic material. What they did, they, as you can see, they and we know that bacteriophages are the viruses which grow, which attack and grow inside the bacterial cells. They infect bacterial cells. So what Hershey and Chase did, they first have grown viruses in presence of radioactive sulfur, some viruses, and other viruses in radioactive phosphorus. As we know that radioactive sulfur, this sulfur is present in protein and phosphorus is present in DNA. So what happened? So the viruses took the radioactive sulfur in their protein, and other viruses took radio-labeled that is P32 radio-labeled phosphorus in their DNA. Then these viruses attacked. They infected the bacterial cells. Then they infected the bacterial cells, and the viruses which were infected with radio-labeled proteins, as we know that coat protein, that is blending, then blending is done, so the protein remained outside. After that, centrifugation is also done. As you can see that radioactive phages were allowed to attach to E. coli bacteria. As the infection proceeded, the viral coats were removed from bacteria by agitating them, as I said, and bacteria infected with virus that have radioactive DNA were radioactive. Means the main thing is, after blending and centrifugation, no radioactivity was found inside the cell which were infected, which have infected radio-labeled sulfur, and radioactivity were found inside the cells which contained or which were infected with radio-labeled phosphorus containing bacteriophages. I repeat, the bacteriophages which contain radio-labeled DNA, which further infected bacteria, those cells shown radioactivity inside the cells. That means we can decide DNA is the genetic material.